Welcome to the Molecular Moments podcast.
In today's episode, we sat down with our guest, Dr. Tina Morris, Executive Director at the American Association of Pharmaceutical Scientists. Tina has a distinguished career in the pharmaceutical industry, including senior leadership roles at US Pharmacopia, Parenteral Drug Association, and most recently, the AAPS. She helped us understand the importance of these industry organizations and how we can make a difference by getting involved. She brings real credibility as a scientist in her leadership roles, as we heard about her experience as a senior level scientist at Ciphergen and Human Genome Sciences. To top it off, we also enjoyed hitting some highlights of science from 2020 and beyond, talking science as scientists do. So without further ado, here's the 5th episode of Molecular Moments. Welcome to the podcast, Tina. I'm delighted that you've joined me today. Can you start by just giving us a few highlights of your career?
Thank you, Chad. It's a pleasure to be here this morning. Thank you for inviting me. I've been with the AAPS since April as the executive director, but I would say my involvement with the organization goes almost my entire career in the United States. I'm a virologist by training, which is shockingly popular this year for obvious reasons. But I would say I've spent most of my career working on proteins. So I have a PhD in molecular virology. I did a postdoc in Tony Fauci's institute at NIH, the NIAID. I worked on hepatitis there. Then I went into industry. You mentioned Ciphergen. That was my first industry job, very small startup company at the time. And then went to Human Genome Sciences, I would say during the heyday of the Genomics era, and then worked at the USP for a very long time, for over 15 years, in a number of different roles. But I would say for the longest time as their head of global biologics, and then most recently as the head of compendial science, before I went to the Parenteral Drug Association as the head of scientific and regulatory affairs for almost 2 years. And this April, I joined AAPS during a very interesting year.
I hadn't realized until I was doing a little background research that you were German, actually. Even having had several conversations, you've managed to lose a bit of your accent. But I'm curious about maybe growing up in Germany and the German education system and how you kind of move into this, into being a scientist coming out of Germany.
I grew up in northern Germany. I'm from Lübeck, which is one of the original Hanseatic League cities. And most people in the world know Lübeck because of one product, that is marzipan.
For sure.
And so everybody who knows me pumps me for it. My mom sends it over for Christmas. So it's a very popular item. But I went to school there. And in Germany, I would say one of the biggest differences is in terms of education that you have to make up your mind about your direction of study when you go to college much, much earlier. So I would say pretty much in high school, at the end of high school, you have to decide, am I going to be life sciences person? Am I going to go into medicine, life sciences, or am I going to, choose liberal arts, history, something like that? And so that's a little bit different. I think in America, the first couple of years of college, I think there's a much, much wider, more broader, different type of education. So I think I had to pretty much decide when I started college that I was going to go into the life sciences. And so I went to the University of Oldenburg, and my bachelor's and master's are in biology. Over those years, I decided I wanted to really go into molecular biology. It was an interesting time. I think when I was in my last year in Oldenburg, PCR hit the big time. And we know that PCR changed everything, right? So by the time I got into graduate school, it was the tool. It was very, very important. Having PCR cloning, sequencing available as tools was a huge game changer. I would say both my undergrad and my graduate experience were different because I did my graduate school training at a medical university. So I switched from a biology background that was very broad at a university that was more focused on, I would say, marine biology, ecological studies, very early focused there on environmental things. That's a very important topic in Germany, has been for a long time. Then switching over into a sort of molecular virology and really changing focus to forever then into sort of more public health and pharmaceutical focus.
When I was thinking about your involvement in USP and PDA and AAPS, that sort of like a civil service of the pharmaceutical industry in a way. We'll come back to those topics in some depth. Was it unusual at that time for women to be going into science in Germany?
I would say yes and no. I was very lucky. I would say that I had good mentors, and I was always surrounded by people, including my family, who were very strong believers and who lived sort of that equality. I also had some great mentors over my graduate advisor in Lübeck, Verena Gauss-Müller. She's a very well-known virologist, a real force of nature, I have to say. She wouldn't mind me saying that. It's true. Everybody who knows her knows that. And that made a big difference. And I think it was also that sort of where I was encouraged to go out into the world. And I think I would say my generation of scientists who were educated at that time in Germany, but in general in Europe, were strongly encouraged and still are to do postdoc work abroad. And as soon as I entered graduate school, it was like, you got to go do something else. You got to go into another country. You got to expand your horizon. And I would say Verena was instrumental in that. I think she was pretty much a trailblazer in her own right, I think, in her own career. And I think if you work with people like that, if you have people like that as your mentors, it's very important, because they give you that confidence that you can go out and do things. And she always pushed us out the door. Go do a poster here, go travel to this, I'll find money, or you can get a fellowship or something. So that was important. That was very important.
One of the things I've noticed in these conversations since I've been doing the podcast now is that the many very accomplished people I've had impeccable timing for their career choices and the fields of study and things like that. And I noticed I think you have as well in some of the directions that you've gone. And I'm interested in your landing at Ciphergen, which I don't think it was bought by someone at some point, but it was really a proteomics focus, I believe. And then Human Genome Sciences, which is of course famous as the Craig Venter organization at the, just when it was at the height of the DNA revolution and the sequencing and all that. So yeah, I'd like to hear more about why you picked those companies and what you're able to do and learn and pick up in those positions.
I guess I was really lucky. I was at the end of my postdoc at NIH, and I was really, even though it was in virology, I was really doing protein science. So I got more and more interested in protein characterization tools. At the same time, I was trying to find another job, because tenure track positions at NIH were very, very hard to come by. And so I can't even remember how many applications I wrote. Then I got a call from Ciphergen, and it was a very small company. They got bought later on. Affinity mass spectrometry platform, very interesting. The inventor of the technology, Bill Hutchins and Bill Rich, were the heads of the company at the time. Bill Rich is of Genomics fame, so he was not new to new technical ventures. Drove out on a dark January morning to be interviewed at BWI Airport by those two guys. And it was a 32-person company with a brand new technology. And that was my first job. And it was very interesting. I was a field scientist, basically. Repaired mass specs, figured out protein applications, worked with a lot of pharmaceutical companies. And as it happened, we did a project at the Institute for Genomic Research. That was Craig Venter's institute at the time. Met some very interesting people there who are, have all moved on in the world now. And then also did a project at HGS. So HGS and Tiger were linked at the time, but then became totally separate entities, where HGS was the commercial drug development company, and then Craig continued on his way down the nonprofit route of which is now the J. Craig Venter Institute. HGS, because of they of course were at the peak of the genome sequencing, gene patenting, discovery phase. We're starting to build up the protein efforts because they realized, after sequencing, once we identify targets, we have to have the proteomics tools. So Bill Heseltine became very interested in the Ciphergen technology because he was hoping that it would be sort of the proteomics complement to what they were doing in gene sequencing. Bill is a visionary guy. I think somebody, I can't remember which reporter, he'll tell you, called him a Renaissance man when they did a profile on him in the 90s. And he still is. If you've listened to my conversation with him on COVID, he's an incredible, incredible person. He wanted to buy into the technology. It was very interesting. That gave me the opportunity to work there. I started working at HGS on that project. And then when they bought into the technology, somehow I got bought with it. So I became an HGS employee at the time to do discovery work with the affinity mass spectrometry platform. And that was incredibly fun. I would say that was one of the most fun times of my career. We got a great publication out of it. It was a very interesting time because so much was happening in drug discovery and so many new tools became available. It was just exploding. And there was a lot of money in the business too. I mean, the late 90s were a fantastic time for biotech. It was incredibly innovative.
Yeah, without a doubt, that was super interesting. I love learning these stories from my guests. Thank you so much for sharing that. Actually, my PhD was affinity mass spectrometry, so I can really, I'm hearing what you're saying. And I worked for MDS in the late 90s and early 2000s. We had the MDS proteomics group, and they were one of the groups that invested. I think they bought something like 30 mass specs to, take that brute force proteomics approach. And that didn't really pan out so much. Why do you think that it worked for genomics and not as much for proteomics?
That's a very interesting question, Chad. I actually spent a lot of time thinking about these things later in my career when I was at USP because of the standardization aspects. And I believe that some technologies or scientific approaches become wildly successful and dominating because there's an early agreement on key items. We did an analysis at the USP when I was there about sort of what are the most important analytical chapters that we have in the USP. And not surprisingly, HPLC was the most important chapter. Now, while HPLC to me is in the same bag as sequencing from the point of view that everybody agrees how it's done and how you get the right answer. And, the key elements are standardized. The way the technology works and the way it can be applied was also at key moments in time widely shared within, the scientific area. Once we got into, I would say, the later days of the genomics game, as you know, once the gene expression profiling started and a lot of these things were done, there was a lot of IP in that game and a lot of things were not widely standardized. So aside from people, and I say that flippantly, right? Aside from people counting red and green dots in gene expression studies, there was no standardization. And there was, at the same time, on a lot of the results, there was a lot of IP. It was not widely shared. And so the field, I would say, was never as impactful because the results didn't have broad comparability. And, what I found out later in my career when I worked on just drug standardization, I found how incredibly hard it is and how much time and how much energy you have to spend on the reagents, the analytical approaches, the statistics, and everything like that. And at a critical time, of opportunity that was never done for proteomics. And I believe that really, really held it back. And it held back the validity of the field, right? And I think it's a huge loss, honestly. And I think, who knows where it could have gone if there had been more sharing, more data normalization, more standards early on. I think that's my personal observation.
My brain's spinning here. And I think, at that time, it was sort of the instruments and computing power. I mean, it was powerful, but felt powerful then. Now it doesn't seem like it was powerful. But so now, instead of that parallel buy 30 mass specs, we can buy one mass spec that has the power of 100 back then, and with the computing power. And so we're making progress now. I think proteomics is certainly more important and maybe making a bit of a revolution with the extremely powerful instruments we have. So it'll be interesting. And as you mentioned, so important to the future. So great segue. You mentioned standardization and where that was a challenge. And you ended up then at USP, where USP is all about standardization. So talk to us a little bit about the mission of USP and what excited you to stay there for 15 years and things like that.
USP is, is the US Pharmacopoeia. It's the official, the standard setting body for medicines that are marketed in the United States. Interestingly, though, is I think the US is one of the very few countries in the world where the Pharmacopoeia is a private organization. It's not part of the government. I think everywhere else in the world, I think one South American country, I can't remember which one now, it's the same, but everywhere else, the Pharmacopoeia is part of the regulator. So USP was unique that way and still is. The mission is obviously to support medicine's quality. One of the most fascinating things to me, which I would say kept me there all these years, was the incredible variety of analytical and scientific challenges that you have to solve. I mean, when you look at, and I don't know the number for the current edition of the USP, but there's way over 4,000 monographs in that book, plus chapters. I think they have a catalog of far over 4,000 reference materials, and each individual drug, anybody who's ever worked in drug development knows you can spend your entire career just figuring out one particular drug, right? And they basically hold the quality keys for thousands of them. And the more you learn, the more you know what you don't know. It's an incredibly challenging but very, very interesting line of work, because if something happens, and it may not even be in your particular specialty area, you have to come up to speed with it very, very quickly. And I was at USP when the heparin crisis happened. And I would say in my, at least, again, very subjective recollection, it feels like from circa 2008 until 2012, 2013, all we worked on was heparin. And it becomes all absorbing. And, you have to just learn everything just to fix a problem. 181 patients died. It became a huge global crisis, a supply chain crisis. It became all absorbing. But you also learn how important it is, right? To have the standards and to make sure that, the raw materials can be tested at the border, that, dialysis patients who get that material, get unfractionated heparin, get safe drug. It's an incredible responsibility.
As a private organization, we put so much trust in that. I don't think people realize it. It's a private organization and we just sort of trust it because we do. How is that sort of maintained with USP? This is something I've never thought about.
Well, part of it is it has an incredibly strong governance mechanism, and the standards themselves are set by independent scientific volunteers who are elected, and the standards are also publicly reviewed. There's a public notice and comment period. The reference materials are tested in collaborative studies. They really, I think, having been around for 200 years now, I think their processes and the governance is very, very strong. Also, I think the way they work and collaborate with the regulator is very well established. So I think the checks and balances really are there to make sure that the standards that go out are scientifically valid and really stand the test of time, which is very, very important.
So the Parenteral Drug Association, you were with them for a few years in a senior leadership role as well. So yeah, let's explore that organization a little bit. Tell me about it, and then we'll hit your current passion and with AAPS, so an organization that I'm deeply involved in as well.
Sure. So I would say the common thread between sort of my last three jobs is working with scientific volunteers. And I think what I learned at USP is how important it is to bring independent scientists together to advance a particular topic independently. And that's true at the USP, that's true at PDA. And at PDA, I was the head of regulatory and scientific affairs. PDA, of course, is all about parenterals. They're very well known for their technical documents, the tech reports. And so basically did a lot of the same things that I did at USP in leading, volunteer teams and developing scientific guidance, commenting on regulatory proposals, doing a bit of policy work there. And I think as you sort of get older and see more of this pharmaceutical science business and regulatory landscape, I think a lot of work becomes also, a lot of my interest also went into sort of policies and how are things done globally. I had done a lot of international harmonization work when I was at USP, working with the World Health Organization, working with other standard setting institutes. And so I have a great interest in that. How does that work globally? And it's always interesting to work at organizations that do have a global reach. What was incredibly fun for me at PDA, which I learned a lot about manufacturing at that organization, because I had always supported manufacturing and CMC, I would say, from the analytical side, but there's such an incredible expertise in manufacturing at that organization. So I learned a ton about sterile manufacturing when I was there, which was great. And it was sort of opened another window in my career to learn something. And I think I'm always happy when I get to learn something.
If there's one thing I've learned about you since the spring when you joined AAPS and I came to start to know you, is that you are always asking questions and wanting to learn. And your leadership in AAPS is clearly both the leader of the organization, but also as a member. That I haven't seen that as much in the past. So I can't stress enough how much AAPS has done for my career over, since early 2000s when I probably first got involved. Just tremendously valuable experience. And I feel like the experience has gotten better the last few years as I've gotten more involved. Well, you wouldn't think too many ways. I'm probably on the edge of being at capacity, but I love it. And I'd like you to talk. Feel free to be an advertisement for AAPS and why you joined and what you want to do. And because I'd love to see the more people we can get involved in this member-driven organization, the better it's going to be for all the rest of us. So
I've been involved with AAPS for a very, very long time. I think I became a member in 98, which is when I joined HGS. One of the greatest benefits to me has always been that I was able to network and learn about other areas of pharmaceutical science and biotech that I didn't know about. And, as we talked a little bit about my career, I kind of stumbled into drug development and in a non-traditional way because I worked for a genomics company. And as soon as we had a drug in phase 1 that I was supposed to support, from an analytical development point of view, I realized I have absolutely no clue about drug development. I'm supposed to do assay development. I really don't know what I'm doing, And so I think at every stage of my career, and especially as my career focus has changed, AAPS has been there where I could just go. I used to go to the National Biotech Conference, which we're bringing back next year, which I'm super excited about.
Me too.
That was just such a tremendous resource for me because the field was evolving very quickly. The demands within our organization were, evolving very rapidly. As soon as you have something in the clinic and you start talking to the regulator, it's like, well, how do I validate a bioassay, or how do I do these things? And it has always been such a tremendous resource for me to go to the annual meeting, now PharmSci 360, to go to the NBC, but also to network with other people in those spaces and really build the network of other scientists that I can talk to. Because if you work in a particular field, and I had worked in protein characterization, mass spec for a while, and this, Chad, the same, the people in that trench. And then all of a sudden it's like, no, you got to do, glycan analysis or bioassay. I'm like, I don't know anybody in that space. I don't know anything about it. I need to find these people. I need to learn so I don't have to start from scratch. And one of the huge, powerful things about our organization is that we have people across the drug development spectrum. We have people in AAPS that work on every kind of modality that you can think of. They're there to talk to. I have to say, once I got to USP, at a very interesting time. We got heavily involved in biologics, but also Roger Williams, who was our CEO at the time, who is an AAPS fellow. He and Vinod Shah and all of these other guys were working on, bioequivalence approaches. And it was a very, very big topic. I learned so much about the expertise that was there and in these areas that I was completely unfamiliar with. And it was very, very powerful because, all of a sudden you have this entire, world of expertise available to you. And especially gaining foothold in that part of my career, I had to learn very, very quickly about modalities and impurity problems and things that from biotech were totally unfamiliar to me, because as my role expanded, I got more and more into small molecule chemistry and CMC, which was not my area at all. And again, APS, deep expertise in that space, lots of people passionate in these different areas. So it's always been a fantastic resource to me. And now even more, I think now that we have those communities online where, any day you can just go on and read what people are discussing about. And you can kind of get a pulse, get on the pulse of what's going on in these different areas in drug development, which I think is fantastic.
Yeah, just this week, actually, I did two things. I joined another community because exactly for the reason that you mentioned, I thought, man, this is an area I need to, build my community, right? And learn, but also it's really hard to learn on your own. And in COVID times, the communities, I think, are more important than ever. So that was cool. And I volunteered to do something else. So so it's just another week in AAPS. So as I said before, AAPS is a member-driven organization. There really is something for everyone. I think sometimes people are maybe intimidated, or they think it's hard to get involved, or they don't know where to start. I'll start by saying, if somebody wants to get involved in AAPS and doesn't know where to go, especially if you're in bioanalytical, look me up on LinkedIn or send me an email or whatever to get in touch, and I'll help you find a place to get involved with AAPS. But Tina, can you talk about what maybe the normal channel would be to get involved with AAPS?
It's that easy, Chad. I mean, some people, even I think people who are not members can read it. I write a column every Friday in the AAPS community, which is an open community where you don't even have to be an AAPS member. You can contact me there. It's the same thing. As you said, you can contact me on LinkedIn. Often people find us because of our conferences or any type of e-learning or educational opportunities that we put on. A lot of people also hear about us because we have very, very strong career development programming and offerings. I think That's always been traditionally also something people are very interested in. Again, sort of the career transitions, graduate students, postdocs, people who are at the sort of decision-making stages of their careers. Should I go into industry? Should I stay in academia? I think we are also unique in that we have really such a great mix of backgrounds. We have obviously a lot of industry folks, but we have a very strong academic representation and regulators, growing presence internationally. And one thing I have always experienced, people are very helpful and friendly. All our members are there. People should not feel intimidated, and members and staff welcome you. Anybody you send an email to at AAPS, including myself, will be happy to talk to you about any opportunities we have for member engagement. It's something that we really take very seriously with every individual member. We realize that this year is very different in the way people interact and the way their work and the way they do their work has been affected by COVID. But I think particularly in that time, the electronic touch points are super important. So, just reaching out and asking the question, and telling us what you're interested in. We want to hear about that too, because we know from the conversations with our members that people over the course of their career seek different things from AAPS. And it's important for us to understand what those things are and what we can provide so that we really have value to our members.
Yeah, Tina, that's a great point, because I can say in my career, I've looked for different things in AAPS, and being at a CRO at BioAgilytix and others in the past. At one point we're a vendor who's doing advertising, if you will, there and looking for new business. And then we're also scientists attending and involved in the organization. So all sorts of different hats. He also made me think about the academic involvement, which I appreciate. And I think, as a active member of the bioanalytical community, we need to get more, maybe this is my pitch, to get more academics involved in that particular community, because it is largely largely a community driven by industry, but there's a lot of really important cutting edge bioanalytical work going on in industry. So can you talk to me a little more about what you're looking at for 2021 with AAPS, with the virtual meetings and whatnot, and how do you begin to sort out? 2020 was hard. 2021 is even harder in this respect.
It's a moving target. What is important to us is to really have a full calendar of interesting offerings. And I'm happy to say that I think we are pretty well planned with great content all the way through August, the Land O'Lakes conferences. But even beyond that, PharmSci 360 just wrapped up, but just this past week, we've seeded all the scientific programming committees for next year. And thank you for leading the bioanalytical Land O'Lakes, by the way, Chad.
You're welcome. I'm excited. It's going to be a great program this year. Yeah.
And what we've really spent a lot of time on during this year is to think a little bit more systematically about our year and our programming during the year. We want to keep open spots available so that we have the ability to respond to what might be going on. You've seen us this year do some very quick turnaround current programming on COVID, but we like to sort of, especially for sort of our cornerstone conferences, make sure that SPCs talk to each other, that they sort of have a common game plan and understand, what's happening at these different conferences, so that our members really have the experience of an ongoing conversation, and that there's different complementary stuff going on each quarter. We're starting in January with another COVID program on immunogenicity, which I'm very excited about. We've got some great speakers there, some great support from the FDA. We have a stability workshop planned for March. And then, of course, the National Biotech Conference. I am super excited. We've got Meena Subramanian and Andy DeGroat as the chair and vice chair, and those ladies are just fantastic. They have very ambitious plans for that meeting. We got a really great slate of programming submissions. I'm really excited about that. And we're planning to pretty much right now be virtual through Land O'Lakes. We're watching, obviously, very, very careful how the vaccine situation is evolving. We're at the same time, of course, already thinking about PharmSci 360 in the fall. I think the expectation is that in the fall we can have a face-to-face component again. So hopefully that'll come out to be true. I think we know that digital will not go away. I think we know that this year has shifted people's expectations, how they learn, how they can get access to content. So I think we're putting a very, very strong emphasis on strengthening our capabilities in that space, beefing up our e-learning tools, and just being able to offer more to our members and making a very, very strong content slate available for next year as well.
I'm excited. I submitted 2 workshop proposals for the NBC meeting. So I hope they both get accepted. If they do, I'm going to certainly have to enlist some help because pulling off 2 of them would be a heavy lift. But there's never, like I said, there's never any shortage of help to find people if you want to, recruit them in to help out. So I'm hoping that those come through. So, I told you we'd talk a little bit about 2020, kind of some highlights of 2020. I started doing research on like the top science accomplishments of 2020, and the first thing that came up was raining lizards in Florida and flying tree snakes. And then I got an email from you that was a little smarter. But actually, on the lizards, it was interesting because I said that it actually is giving, they actually are studying those lizards because they froze and then fell out of the trees, right, in the winter in Florida. And they said actually they're now using it to study how climate change may or may not affect the reptile population. So it was actually a legit research. It was pretty interesting. They said they're much more durable than they thought they would be because some of these things froze solid and fell off the trees, and then when they melted or thawed, they ran away. And the snakes, I think it was more about like the aerodynamics of the snakes and things like that. It was actually pretty interesting. But I think you were more on the right direction when you emailed me, and I can't think of 2020 in science and not think about the Nobel Prize in Chemistry with Jennifer Doudna and Emmanuel Charpentier with CRISPR. That was super exciting. And I just talked with Binod da Silva, a fantastically accomplished woman in science. And I'm speaking with you, have had a fantastic career in science. And these 2 ladies did so much. So, I mean, what did that mean to you as a woman or just as a scientist, right? Reacting to that Nobel Prize?
As you said, super exciting. And I'll put out a teaser here that, we were, of course, super lucky and fortunate that we had already booked Jennifer Doudna as our plenary speaker for PharmSci 360. And she graciously also agreed to do an interview with us for the AAPS news magazine. So Gopi Shankar, our board member, and myself got to interview her afterwards, which was honestly super inspiring. And so that was definitely one of the highlights of my year. And, you can look forward to reading some of her insights in the news magazine, I think in January. But what I was excited about, and we talked to her a little bit about that, is that they did not wait for, decades basically to award the Nobel for this discovery for CRISPR, which I found very interesting because when you look back on some of the others that were given for sort of molecular biology milestones like PCR, it took much, much longer. It's going to be very interesting to follow what that does to the field and, how can it accelerate the development in that area. And I think that's terrific. And I think she's just a really inspiring person. What was really interesting to learn from her also, and that was, touches back on what we talked a little bit about earlier, is she has 1 foot in academia and 1 foot in industry, right? Because she co-founded several companies. She's very involved on the industry, on the drug development side. But at the same time, she's still pursuing her academic career, and she's also involved in the in the ethics considerations of gene editing, which of course are also very, very important, right? This is something I think that'll keep people discussing for quite some time. So I think this was very, very important. That was probably the important thing this year scientifically. I'm very excited about it. If you ask Joy, by the way, Joy Davis, I'll give a shout out to her, our managing director. She thought this was going to be the year of the murder hornets, by the way. And that didn't play out. So,
No.
But I think we got almost everything else this year, but not the murder hornets. So,
what really stuck out to me, so I had the good fortune, and thank you for the invitation to participate in the small group discussion with Jennifer. And I can't say enough that, to agree enough that how inspiring she was. And the other thing that stuck out was she commented that, one of the things she wants to do with her prize money is to really put it towards underrepresented groups in the sciences, which is something that I feel strongly about as well. And I thought, goodness, this is one of those people who seems to have 30 or 35 hours in her day while the rest of us only have 24. I don't know how she can accomplish and do everything that she does. So without a doubt, that was one of the highlights. So, maybe we'll touch on one or two other highlights of 2020, scientific highlights. Is there something else that jumps out to you that was really exciting from 2020?
I think obviously everybody has their, I would say, interest window or, things they follow. I just feel like there's been so much. Maybe it's because coming to AAPS and I had the privilege of working on a lot of different topics this year, but we spend a lot of time working on nucleic acid-based therapies. This is something, beyond CRISPR and gene editing. That's an area that's picking up incredibly. Part of that, I think, is just the incredible formulation science that's going on. Again, not just the COVID vaccines, but these lipid nanoparticles, the packaging for these materials, the molecular science that goes into that, the new lipids that are being discovered, incredible. I found that very interesting. We did a custom workshop on oligonucleotides, which kind of immersed me into sort of the entire history of that drug class and really understanding how now just over the last few years, these enabling technologies make that, a drug class to have huge potential. But what they had to overcome to get there, and that's been fascinating to me. And I look forward to an entire track on that at the NBC. Now, from my work at USP, I continue to be very interested in impurities and drug quality and supply chain. And of course, sort of one of the add-on effects of the whole COVID situation, of course, is that it's disrupted the entire global pharmaceutical supply chain in a massive way, right? I've worked a lot with generics side of things, and global supply chains have been challenged. And, we've had new impurity challenges, the nitrosamine issue, many other things. Those are without a global pandemic going on. These are complicated things to tackle. But I would say particularly in a year like this, it's been very interesting to see how the industry has adapted to the situation and what's been necessary to tackle some of these challenges, because we've seen drug shortages pop up for drugs that hadn't had shortages in years, right? And so it's been very interesting following that and to see how the industry adapts to deal with this public health challenge.
And of course, when you talk about nucleic acid therapeutics and when we talk about CRISPR, which the CRISPR technology has led to tremendous advances in delivery mechanisms with viral capsids and whatnot, which has really enabled the 2 vaccines that we now have. Super, super exciting, and how all those different fields interrelate. Well, it takes a village in science as well. So I want to touch on one last thing, that just a little bit of fun before we wrap up. So, I've been asking all my guests this year, what sorts of hobbies and things is there anything new you've taken on in the midst of COVID to keep you busy?
I would say I've taken on something new, but I've probably done more of what I've already liked to do. I'm a knitter. I craft, so I enjoy knitting. My other hobby is photography. Obviously, my photography has been much more local than it has been in the past. I've been blessed over the course of my long career that I traveled extensively. So I've done a lot of travel photography over the years, and it's been a lot more time spent on post-production of photos and things that have been created in years before. We've also, both my husband and I, enjoy cooking. We always set a goal to try a certain number of new recipes. And because we've been at home, we've exceeded that goal by quite a few new recipes. So that's been fun.
Yeah, well, that's fantastic. If you could see my office behind me, but if you could see the rest of my office, you'd see that actually, while it's my office, it's also my wife's yarn storage room. And it's sort of wall to wall yarn. Yeah, something I've learned as she's been a knitter for 25 years or so, but something I've learned is knitters collect a lot of yarn and they never get rid of the old scraps. So
yes.
So someday, hopefully you can meet her. I'll bring her to an AAPS meeting or something, and that'd be fantastic. So the other thing we were joking about before is you and I both have hoodies.
Yes.
And as you said, this is definitely a hoodie year. So it's been fantastic talking to you. Every time I do these discussions, I feel like we unpack so many things. I could spend 2-3 hours doing these interviews. They could be like a Joe Rogan, 2 or 3 hour interview. But I do want to, thank you again for joining us. I'm so excited about what you're doing at AAPS, and it's so great to get to know you better. Looking forward to sharing a glass of wine with you sometime this year. It will happen. I'm confident of that.
That'd be great.
Do you have any other closing comments or thoughts for us as we close out?
No, I just want to thank you very much for inviting me, Chad. It's been a pleasure. I'm a people person, so having come to AAPS and, obviously I already knew a lot of people when I came, but it's been a great pleasure getting to meet so many of our members and, collaborating across the scientific spectrum. And that's really something I tremendously enjoy. And I just want to, encourage everybody who might be listening to check us out. We're a friendly bunch. The worst thing that can happen to you is that you'll be volunteered for something that is going to be fun. So yeah, it's been a great pleasure being on and chatting with you. So thank you for inviting me. Hope that our listeners get interested in checking us out.
Tina, thanks so much. So that is all for episode 5. If you enjoyed today's episode, be sure to subscribe on Apple Podcast, Spotify, or your favorite podcast app so you never miss a conversation. If you'd like to hang out with us outside of the podcast, we have many webinars and other presentations available for your enjoyment and education. Visit bioagilytix.com to see what's coming up and how you can stay in touch. And since we talked a lot about AAPS today, I'll add visit aaps.org and you'll find a lot of material there as well. So don't forget to keep an eye out for season 2, which is going to roll out in early 2021. We're looking forward to some great guests. We'll have world-renowned experts talking about gene therapy, diversity in the pharmaceutical industry, new and exciting technologies coming, and a conversation with a patient who's benefited from some of the recent tremendous developments in our industry. Molecular Moments would not be possible without the support of our sponsor, BioAgilytix Labs. BioAgilytix is a global contract research organization specializing in large molecule bioanalysis. Based in Durham, North Carolina, with labs in Hamburg, Germany, and Boston, Massachusetts, BioAgilytix provides high-quality bioanalytical services to leading pharma and biotech companies around the world. They offer assay development, validation, and sample analysis under non-GLP, GLP, and GCP, as well as GMP quality control testing. If you're looking to work with a team of highly experienced scientific and QA professionals through all phases of clinical development, look no further than BioAgilytix. For more information or to speak with their scientists today, visit their website at www.bioagilytix.com.
Thanks for listening to the Molecular Moments podcast.